As we discussed earlier in the course, there are some major anatomical structures in the brain that are easily identified on the basis of being physically separate from other structures, having different types of neurons, different physical arrangements of neurons, and different patterns of connectivity. These are the most prominent, and we'll be describing each in turn. The cortex and the fairly similar hippocampus, various subcortical structures like the thalamus, the basal ganglia, the hypothalamus, and the basal forebrain, the amygdala, that's a bit like the cortex and a bit like the subcortical structures, the cerebellum's a bit like a separate cortex plus subcortical structures. It's located below the back of the main cortex, and a number of other structures down in the brainstem, in many ways resembling the subcortical structures higher up in the brain. We've just finished a description of the underlying neurophysiology and chemistry, and even if it was an overview, we still covered a lot of material. And now we're going to plunge into a description of all sorts of anatomical structures. And as we described earlier, there's a range of techniques by which information about these structures has been acquired, in total amounting to a vast amount of information. So, how on earth can we make sense of all this material? Well, once again, this is the diagram I showed earlier. It comes from later in the course, and it's going to be the foundation for tying all the cognitive and anatomical information together to create the hierarchies of description we need, because it links all the different anatomical structures to the information processes they perform. At this point, we're going to go through anatomical information about the major structures. Then, after we've looked at how natural selection has constrained the brain into this architecture, we'll review the anatomical information and see how it demonstrates that these are the information processes being performed. But one important thing to bear in mind is that major brain structures perform information processes. They don't correspond with obvious cognitive processes. And an example I touched on earlier, recording and accessing memories of events, which were called episodic memory. During episodic memory processes, activity has been measured in all of these major structures. Damage to an even wider range of anatomical structures results in episodic memory deficits, presumably demonstrating that there's activity in those extra structures as well, but it's not detectable by current techniques. So we're aiming to understand what exactly it is that all these structures contribute in different ways to cognitive processes like episodic memory. So we'll begin with the most visually obvious structure, the cortex. The cortex is this thin sheet of neurons, 2 or 3 millimeters thick, wrapped around a large volume that's mostly the axons of cortical neurons. Now, over 99.5% of these axons just connect between different cortical neurons, and a very small proportion go to other anatomical structures. The cortex has an area about the same as a couple of sheets of paper, but it's crumpled up to fit inside the skull. The neurons in the sheet are organized into a number of layers. Conventionally, we count 6 layers, numbered by the Roman numerals 1 to 6, with layer 1 on the outside closest to the skull. About 85% of the neurons in the cortex are a type called pyramidal because of the shape of their somas. Their axon emerges from the base of the pyramid. An apical dendrite emerges from the opposite end of the soma from the axon, and a number of basal dendrites emerge all around the soma in lots of directions, but all perpendicular to the line defined by the axon and the apical dendrite. Pyramidal neurons use the neurotransmitter glutamate, so they're classified as excitatory neurons. And pyramidal neurons also occur in the hippocampus, which, as I mentioned, is in some ways an extension of the cortex. And similar neurons also occur in parts of the amygdala, but they're not found in other structures. On average, each pyramidal neuron has inputs from about 10,000 other pyramidal neurons. The synapses are located on the tips of spines that stick out from the stem of the dendrite, and they release glutamate to excite the target neuron. And pyramidal neurons also get inputs from inhibitory neurons, and these inhibitory neurons are also located in the cortex, plus they get inputs from other anatomical structures, such as the dopaminergic inputs that play a role in changes to the synaptic weights of the glutamate synapses, as we discussed earlier. Now, this illustrates some different types of neurons in the cortex, and the positions of these neurons relative to the 6 cortical layers are also indicated. The layers are shown on the left. The physical appearance of cortical neuron dendritic trees can vary considerably, and as you can see, much of the variability is between neurons located in different layers. But underneath all this variability, it's possible to distinguish 2 major types of neuron on the basis of 3 characteristics. The 2 types are pyramidals and interneurons. The first of the characteristics in which pyramidals and interneurons generally differ is that the dendritic trees of pyramidal types have a spiky appearance in a microscope because they have lots of spines. Interneurons look smooth. The second characteristic is that pyramidals are excitatory, using the neurotransmitter glutamate, while interneurons are inhibitory, they use GABA. Thirdly, interneurons only target neurons in their immediate vicinity, often only within 500 micrometers, half a millimeter. Most pyramidals are projection neurons, meaning that they target other neurons at long distances away. Neurons that project the longest distance tend to be larger. The pyramidals in the motor cortex that project all the way down to the spine are particularly large. The one exception to these rules is the spiny stellates, that are small pyramidal neurons but only project in their immediate vicinity. Now, cortical resources are organized in an apparent hierarchy. There are large numbers of pyramidal neurons that have a general similarity to each other, although they differ in detail. The dendrites of pyramidal neurons have a number of terminal branches, and these terminal branches again are generally similar. Then terminal branches are made up of groups of synapses that again are generally similar but differ in detail. And going the other way, pyramidal neurons are organized in columns, with rather more connectivity within a column than between columns. And the cortex as a whole is separated into areas, again generally similar to each other but with detailed differences. So multiple similar structures can be seen on a number of levels of detail in the cortex. There are large numbers of pyramidal neurons, all of which have similar form but are individually different at a more detailed level. There are multiple layers, but the layers differ. There are many columns, but again some differences between individual columns. There are a number of areas, more than 100 in fact, that are similar but can be distinguished by detailed differences in the type of layering and various neurochemical differences. So in the next few sections, we'll have a closer look at the anatomy on some of the higher levels.